Have you ever really stopped to wonder what a memory is? I mean, physically, when you learn something new, a simple fact, a life-changing event, a real, actual, tangible change happens inside your brain. But what is it? And where is it? For more than a century, scientists have been on this incredible quest to find the physical trace of memory. And today, we're going to join that hunt.
Now, this question sounds so simple, right? But it is one of the deepest, most profound mysteries in all of neuroscience. We all know memories are in the brain, but if we could just zoom in with some impossibly powerful microscope, what would we actually see? Would it be one special cell, a specific molecule, a unique pattern of electricity? The search for this physical trace of memory, well, that's the search for what scientists call the engram.
The word engram was created to describe this exact idea, this hypothetical trace. As some of the top neuroscientists in the field, Sheena Jocelyn and Susumu Tonegawa, put it, the very fact that memories last implies that some kind of physical, internal representation of this experience is stored in the brain. The engram is that representation. It's a specific, lasting change that's created by experience. And today, we're going to see the unbelievable tools scientists are now using to finally see it and even control it.
So, here's our roadmap. We're going to follow the exact, elegant logic these researchers used. First up, we'll see how they create a super precise memory in the lab. Then, how they actually tag the specific brain cells involved in that exact moment. After that, we'll find a correlation, and then, and this is the really cool part, comes the ultimate test. Can they turn that memory on command? Finally, we'll look at how these memory cells actually wire themselves together and even link up with other memories. It's going to be quite a ride.
All right, Section 1, the memory trace. Before you can even begin to find a memory, you have to create one. And you can't just use any old memory. To do this scientifically, you need one that's super simple, completely reliable, and totally measurable. This control is absolutely everything, because it lets researchers know the exact moment a memory is formed, and that is the critical first step to finding its physical address in the brain. The classic method for this is called contextual fear conditioning. It's a really straightforward but incredibly powerful way to learn. So here's how it works. First, a mouse gets to explore a unique little environment. We'll just call it context A. Then, at some point in that context, it gets a mild but unpleasant foot shock. The mouse, of course, instantly forms an association, a memory that links this specific place with that bad feeling. So what happens when you put it back into context A later? Well, we can measure its memory by its natural fear response. And for a mouse, that response is to freeze in place. The longer it freezes, the stronger we know that memory is.
So with this process, we now have a specific, measurable fear memory locked and loaded. The memory is reliably formed, which means the hunt for the engram cells that are actually holding it can finally begin. Okay, so the brain just formed a memory of context A. Billions of neurons are firing away, but which tiny fraction of those are actually storing that specific memory? The huge challenge was figuring out how to shine a spotlight on only the neurons that were firing most intensely at the exact moment of learning.
The breakthrough came from this really special class of genes called immediate early genes, or IEGs. Honestly, the best way to think about these is like cellular flares. When a neuron is firing like crazy, like it would during a surprising or really important event, it quickly flips on these IEGs, like CFOS and ARC. And this activation leaves behind a biological marker that basically screams, hey, I was just super active a second ago.
And this is where the pure genius of modern genetic engineering comes into play. Scientists developed a technique called activity-dependent tagging. They basically hijack the on switch for an IEG like CFOS. Then they link that switch to another gene that they've artificially put into the mouse's DNA, a gene that, say, makes a glowing green protein. So what's the result? Well, only the neurons that fire intensely during that foot shock will flip the CFOS switch. And when they do, they turn themselves permanently green. This tags them. These are now our prime suspects for being the engram.
Okay, so we've tagged the neurons that were active during learning, but is this really the engram? The very first test is to look for a correlation. When the mouse is put back in that box and naturally remembers the shock, do these exact same tagged cells light up all over again? To test this, researchers used a really clever control group. The paired group is the one we just talked about. They learn that context A is associated with a shock, so they form a strong memory. But the critical, unpaired control group gets the shock in context A, but without any prior experience of that context. So for them, the shock is just this random, surprising thing that isn't really linked to the place itself, and because of that, they form a much, much weaker memory of the context.
And when we look inside their brains, wow, the difference is just striking. These are actual slices from two key memory areas: the hippocampus, which is vital for remembering places, and the amygdala, the brain's fear center. Those little green dots, they mark the neurons that were reactivated during the memory test. I mean, just look at the difference. In the paired group on the right, those are the animals with the strong memory, you can clearly see so many more green reactivated neurons in both brain regions. And this chart just gives us the hard numbers. It confirms what we just saw with our own eyes. In that paired group, a much higher percentage of neurons were reactivated. This right here is our first critical piece of evidence. It's a direct correlation. The stronger the animal's memory, the bigger the population of these reactivated neurons. But look, a correlation, no matter how strong it is, isn't proof. To prove that these cells are the memory, scientists have to clear two very high bars: necessity and sufficiency. Necessity means that these cells must be active for the animal to remember. You could test that by silencing them and seeing if the memory goes away. But the even more dramatic test is for sufficiency. Is activating only these cells enough to force the animal to recall the memory, even when it's in a completely safe, totally unrelated place?
To do this, they used a revolutionary tool called optogenetics. It is literally like installing a light switch directly onto a neuron. So instead of tagging the Engram cells with a glowing protein, they used that same IEG trick to insert a gene for a light-sensitive protein called an opsin. Now, these specific Engram cells are the only ones in the entire brain that will fire when you shine a blue light on them. By implanting a tiny fiber optic cable, scientists gain this unbelievable ability to turn these memory cells on whenever they want.
And this sets up the single most critical experiment in the whole field. Okay, so the mouse that formed the fear memory back in context A is now placed in a brand new, totally safe environment, context C. It has absolutely no reason to be afraid here. So what happens when the scientists just flip the light switch and artificially reactivate the brain cells that are associated with that scary place? And the result is absolutely stunning. You can see it right here on the graph. In this new, safe context, the mice are just behaving normally. But the moment the laser is turned on, activating only those tagged memory cells, the mice with the light switch protein, that CHR2 group, they instantly freeze in fear. They are behaviorally recalling the memory, even though there are zero external reminders of the scary place. This is the smoking gun. It proves that this specific group of neurons isn't just involved with the memory, activating them is sufficient to trigger the entire memory recall. These cells, they are the physical trace of memory. Okay, so we found the Engram cells, and we've proved what they are. I mean, that is an incredible achievement. But a memory isn't just a random pile of cells. It's a precisely connected network. So the next logical question is, how do these specific neurons get wired together to form one single, coherent memory trace? What's the physical glue that holds them all together? Well, the answer lies in a principle that was proposed all the way back in 1949 by a guy named Donald Hebb. It's famously summarized as neurons that fire together, wire together. This idea, which we now call Hebbian plasticity, is pretty simple at its core. When one neuron helps to make another one fire, the connection, or the synapse, between them physically gets stronger. This strengthening is learning at its most fundamental level. And this isn't just some abstract theory. We can actually see these physical changes under a microscope. During memory formation, the connections between these Engram neurons go through real structural changes. They can grow brand new physical contact points, and the connections that are already there get chemically strengthened in a process we call synaptic plasticity. All of this makes sure that the specific group of neurons is now wired together, making it way easier for them to fire as a coordinated team the next time the memory is recalled. Let's zoom right in on the key mechanism for this strengthening. It's called long-term potentiation, or LTP. When the Engram neurons are firing at a really high frequency during learning, the neuron on the receiving end physically inserts more listening posts called AMPA receptors right at the synapse. With more of these receptors, it becomes much more sensitive to the signal from the sending neuron. This makes the connection powerful and really long-lasting, which is the very definition of a physical memory trace.
But of course, our memories don't live in a vacuum, right? We are constantly linking new experiences to old ones. If you try a new coffee shop, your memory of the coffee gets linked to the memory of the street it's on. So how does the brain physically wire these separate memories together? This slide here illustrates the leading theory. When two different things happen close together in time, their engrams are way more likely to be formed by an overlapping group of neurons. A subset of brain cells actually gets recruited into both the engram from memory A and the engram from memory B. This co-allocation of neurons, this shared cellular hardware, is thought to be the physical basis for how we form associations. We have come an incredible distance today, from this purely hypothetical idea of a memory trace to being able to activate a specific memory with a literal beam of light. But for every neuron that gets excited and gets to join an engram, there are many others that are actively being quieted down. And this leads to one of the next great frontiers in this field. What is the role of the brain's traffic cops, the inhibitory neurons whose entire job is to tell other cells not to fire? How do they help choose which lucky few neurons get to become part of a new memory? Answering that question, well, that will surely open up a whole new chapter in our quest to understand the mind.